Hello dear children, welcome back. I'm Dr. Jisha Joe. In this session, we shall study about the structure and function of mitochondria. Now coming to the discovery of mitochondria were discovered by Fleming and Colliker in 1882. But the term mitochondria was put forward by Benta in 1889, and the presence of respiratory enzymes in the mitochondrion were discovered by a person named Warburg in 1913. So once again, mitochondria were discovered by Fleming and Colliker in 1882. The term mitochondria was put forward by Benta, and the presence of respiratory enzymes in the mitochondria were discovered by Warburg in 1913. Mitochondria are cell organelles present in both plant cell as well as in animal cell. And if we consider the shape of mitochondrion, it is sausage shaped, or we can say it is a rod shaped or cylindrical in shape. Okay, now coming to the structure of mitochondria, as I told you, it is oval in shape, or it is sausage shaped or cylindrical in shape. That is the mitochondrion. And it is about 0.5 to 1 micrometer in diameter. 0.5 to 1 micrometer in diameter is the size of mitochondrion. It is oval in shape, or it is cylindrical in shape, we can say. It has two membranes. It is a double membraned structure. The outer membrane, that is the outer membrane. The outer membrane is a freely permeable membrane, and it is made of proteins called porins. Porins are the proteins that are present in the outer membrane, and the outer membrane is a freely permeable membrane, and it is permeable to small molecules and certain ions. So that is with regard to the outer membrane. We know mitochondrion is rod shaped, and it has two membranes. The outer membrane is a freely permeable membrane. It is a freely permeable membrane. It allows small molecules and ions to pass through it. It has proteins called porins present on it. Now when we move to the inner membrane, that is the inner membrane, which I am going to draw in another color. The inner membrane has several infoldings. Can you see the infoldings there on the inner membrane? So these infoldings are present on the inner membrane. Okay, these infoldings are called as the cristae. Cristae. Okay, so and that is the inner membrane. Inner membrane. So it has two membranes, the outer membrane as well as the inner membrane, and the two membranes are separated by a space which is called as the perimitochondrial space, or it is the intermembrane space. That is the space here. That is the space that you see in between the two membranes is called as the perimitochondrial space. Perimitochondrial space, or it is called as the intermembrane space, the space between the two membranes, intermembrane space. It is about 80 to 100 angstrom width. So once again, the mitochondrion is an oval shaped structure, or it is sausage shaped or rod shaped, we can see. And the mitochondrion has two membranes there, the outer membrane and the inner membrane. Outer membrane is continuous, which I have drawn in black. That is the outer membrane, and it is made up of proteins called porins. It is a freely permeable membrane, allowing small molecules and ions to pass through it. Now coming to the inner membrane is also continuous, but it has several infoldings which we call as the cristae. The infoldings of the inner membrane of the mitochondrion are called as the cristae. Okay, and the space between the two membranes, the outer membrane and inner membrane, is separated by a space which is called as the perimitochondrial space, or otherwise it is called as the intermembrane space. Now, the inner membrane, that is the inner membrane which I have drawn in red, that encloses a space inside. This space is filled with a matrix which is called as the mitochondrial matrix. It is a fluid which is called as the matrix. Okay, so the inner membrane encloses a fluid matrix. This space, and in this space which is enclosed by the inner membrane, there is a fluid which is called as the matrix. Now, once again, coming to the inner membrane, the inner membrane is not a freely permeable membrane as we have seen in the case of outer membrane. It is a selectively permeable membrane, and it is permeable only to oxygen and ATP. The inner membrane is permeable only to oxygen and ATP. It is a selectively permeable membrane. And the matrix contain the enzymes for the Krebs cycle. Krebs cycle is a phase in respiration, and all the enzymes that are needed for the Krebs cycle are present in the matrix. Okay, so I hope that is clear. Then moving on to the inner membrane. If you look closely at the inner membrane will have several stalked particles called cristae. Stalked particles called oxysomes will be present on the inner membrane. You can see the stalked particles throughout the inner membrane. Throughout the inner membrane, you can see stalked particles. So I am just drawing only a few, but we can see the stalked particles which are called as the oxysomes. Throughout the inner membrane, you can see the oxysomes. Okay, throughout the inner, I've just drawn only a few, but oxysomes are stalked particles. They are found distributed throughout the inner membrane. Now, moving or looking closely onto the structure of the oxysomes. Oxysomes, as I told you, they are stalked particles. They have a base. There is a base there. There is a small stalk, and a head region is present. So that is the structure of an oxysome. Okay, it has a basal region, that is the base, that is the stalk, and that is the head region. So that is the structure of an oxysome. It is otherwise called as the F0, F1 particle. The stalk head piece is otherwise called as the F1 particle, and the base is called as the F0 particle. Okay, F1 particle is the head and F0 particle is the base, and so oxysomes are also called as F0 F1 particle or F0 F1 ATPase. That is because they are concerned with the synthesis of ATP. Okay, so that is with regard to the oxysomes. So once again, moving on to the structure of mitochondria. Mitochondria is sausage shaped or rod shaped or cylindrical in shape, and it is about 0.5 to 1 micrometer in diameter, and it has a it is a double membraned structure. It has two membranes. The outer membrane is a freely permeable membrane which is made up of proteins called porins, and it allows small molecules, ions, etc. to pass through it. Then inner membrane is called called, the inner membrane has several infoldings called as the cristae. The inner membrane is a selectively permeable membrane. It allows only oxygen and ATP to pass through it. Then when we look more closely onto the inner membrane, you can see certain stalked particles. These stalked particles are called oxysomes. Stalked particles. Since it has a stalk, it is called as the stalked particles. These are called as the oxysomes. Coming to the structure of the oxysome, each oxysome has a base, a stalk, and a head piece. The base is called as the F0 particle, and the head is called as the F1 particle. And so oxysomes are also called as the F0 F1 particle or F0 F1 ATPase, because it is concerned with the synthesis of ATP through the process of oxidative phosphorylation. So that is with regard to the oxysomes. Now the inner membrane encloses a space inside, that is the space which we see here. So this space is filled with a fluid and it is called as the matrix. All the enzymes that are necessary for respiration, especially for the Krebs cycle, respiratory enzymes for the Krebs cycle are present in the mitochondrial matrix. The mitochondrial matrix, as we have seen, it also contains DNA and ribosomes in it. Ribosomes present in the mitochondrial matrix are the 70S ribosomes. So, presence of DNA inside the mitochondria enables the mitochondrion to multiply, and this process of replication, it is by fission. Multiplication of the mitochondrion occurs by the process of fission, and it occurs during the G2 phase of the cell cycle. G2 phase, the mitochondria duplicates. The DNA inside the mitochondria duplicates. The mitochondria also duplicates, so that after the next stage, that is after the cell division, nuclear division, the mitochondria get distributed to the two daughter cells. So mitochondrial matrix has the DNA, and the ribosomes present are the 70S ribosomes. The presence of the DNA or the genetic material enables the mitochondria to duplicate, and this duplication of the mitochondrion occurs during the G2 phase of the cell cycle. So that is with regard to the structure of mitochondrion. Now moving on to the functions of mitochondria. As we have already mentioned, mitochondria are the sites of cellular respiration. We have already seen all the enzymes, respiratory enzymes that are necessary for the Krebs cycle are present in the mitochondrial matrix. And also the stalked particles, the F0, F1 ATPase that are present on the inner mitochondrial membrane. So on the inner mitochondrial membrane, it is the site for the electron transport chain. And finally, the presence of the F0, F1 particle on the inner membrane helps in the synthesis of ATP. So mitochondria are the powerhouses of the cell. We study such a term in lower classes. Mitochondria are called as the powerhouses of the cell because they are concerned with the synthesis of energy in the form of ATP, and it is because cellular respiration takes place inside the mitochondria. So that is with regard to the functions of mitochondria. So in this video we have seen the structure, the discovery of mitochondria. We have come to the structure of mitochondrion, and finally we have also discussed about the function of a mitochondrion. Children, I hope this session is clear to you. If you have any doubts or suggestions, please ask your doubts and give your suggestions in the comment section. Thank you.